Hi everyone, I'm Lamia Abdelazim from UC Berkeley, and I'm excited to kickstart this Flash Talk session and give you a snapshot of my research as a postdoc in the DESNIC lab developing next generation optical interfaces. So general principle of neuroscience is that information from the external world is encoded into the brain, and that these neural codes are what generate all forms of mental experiences. That is the case in particular for perception and vision. These neural codes are implemented by neural circuits, with the single neuron and the single spike or action potential being the basic units of computation. On top of that, visual circuits are distributed across multiple brain areas that are thought to be organized in some form of hierarchy, with the main stream of information traveling from the retina up into higher cortices. But the interactions actually between these areas are much more complex, since there's also information traveling from top to bottom, as well as within each area. So let's say we want to recreate vision. State-of-the-art electrical prostheses can only generate phosphene percepts, which look like these blurry dots of light. To recreate more naturalistic patterns, we need technologies that will allow us to access these interareal circuits and directly tap into these neural codes with high spatial and temporal resolution. For single-cell resolution read-write, we use two-photon calcium imaging to record population activity and combine it with optical holography. By shaping the phase of our light beam, holography enables us to have a user-defined pattern at the microscope objective, which then allows us to activate individually selected neurons of interest. But so far, what has been achieved with two-photon holography is near binary on-off control, as you can see in this plot showing neural activity as a function of time. The patterns are very similar across the set of activated neurons. To go beyond this limitation, we first developed a new viral construct, merging a potent opsin Cron2F with a fast calcium indicator, G-Combite-M. To get full 3D, we then built a new microscope that combines 3D holography with high-speed volumetric Bessel imaging, which records a full 3D volume in a single-shot 2D projection. With all these combined, we were able to read and write in full 3D precise optogenetic spike trains with single spike precision up to the Nyquist limit, as you can see in this trace example. With this, we can leverage this platform for high replay fidelity. Here you can see an example where I would record neural activity as I present a movie to the mouse, and then holographically replay the exact spatial-temporal pattern with very high fidelity. Finally, we want to achieve interareal access. So another project I led during my postdoc was the development of a holographic two-photon mesoscope that allows us for the first time to do pattern photostimulation in one area, while simultaneously recording neural activity from several areas around and with single-cell resolution. So with this new platform, we demonstrated that we can record long-range postsynaptic responses to optogenetic stimulation, sometimes millimeters away. Finally, we did a number of perturbation experiments where we stimulated area LM in the mouse visual cortex with distinct holographic patterns. After training a decoder on other areas, excluding area LM, the decoder was able to successfully discriminate the holographic stimuli above chance level, indicating that we can holographically add information in the network that gets transmitted in downstream areas. To summarize, I've shown you here that we now have the technology to mimic endogenous neural activity with very high fidelity in full 3D with single cell resolution and single spike precision, and that we can generate pattern perturbation in one area and simultaneously measure downstream effects in multiple surrounding areas. So building on these techniques, my future research directions will be, on one hand, to interface these optical microscopes with predictive models of neural activity to design end-to-end cortical prostheses, and in parallel to investigate the interareal codes of perception that underlie the construction of comprehensive and holistic percepts. With that, I would like to thank my mentor, Hillel at DESNIC, as well as my colleagues in the lab and our collaborators and funding. Thank you for your attention, and I'll be happy to take questions during the Q&A session. Thank you.
Hi, everyone. My name is Christine Chua, and I'll be talking about Filipino cultural values and how they shape responses to stress in a cohort of pregnant Filipino women. So fear and concern about the current social political environment are becoming a worldwide issue for maternal and fetal health. So this is a map of the 2021 Global Peace Index. And as you can see, the areas with more conflict are presented in red and orange, and they tend to be countries in the global south as well as Russia. There's been some evidence suggesting that the political climate might impact pregnancy outcomes in negative ways. And so, for instance, in the US, there's a recent report documenting the health effects from anti-Muslim discrimination following the 9/11 attack on the World Trade Center. And so not only have Arabic American women been experiencing elevated rates of preterm birth and low birth weight, but other women who tend to be misidentified as being Muslim also have shown similar patterns. And so it's unclear, however, how the social political stress might become embodied to later affect maternal and fetal health. And so I set out to investigate this question in a cohort of pregnant Filipino women. So one of the main motivations for asking this question in the Philippines was because of the war on drugs that was initiated by former President Rodrigo Duterte. So in his war, he incentivized extrajudicial killings of suspected drug dealers and users who failed to surrender to local officials. So in this study, I conducted a series of virtual interviews from 21 pregnant and recently pregnant women from all over the Philippines in 2021. And this was to gain a better understanding of how these women's experiences were with the war on drugs. So here the yellow stars with numbers represents the number of women recruited from each region. These women were around 28 years of age, having had more than one child, and mainly were from low to middle socioeconomic backgrounds. Roughly half were pregnant at the time of the interviews. So what was striking was that there was almost a 50-50 split on whether they approved or disapproved the drug-related killings, and that these interviews showcased nuanced reasoning, with some stating that they approved of the war on drugs, but not how they were carried out. So what I'd like to offer here is an example of a translated text where this woman describes how she's managed her own stress levels. So here she says, "Right now, the war on drugs doesn't affect me much. Although I know it's still ongoing, there are still stories about it, but it's not the focus of the media. I don't read enough information about it. And lately, especially now that I'm getting close to giving birth, I have been trying to disassociate myself from negative feelings, although they're realities and they're happening." So what's interesting about her response is that it highlights the Bahala culture in the Philippines, which means that you leave everything up to God or fate. And this is a culture-bound attitude that many will invoke to downplay their experiences of stress. And it's suggesting what I think to be considered a coping strategy, but it also shows that women may not be perceiving themselves to be directly impacted by the Philippine politics. So based on these interviews, there were three major themes that emerged, all of which contributed to maternal perceptions of the social political climate. So here I used the iceberg model of culture to visually demonstrate how deeply rooted cultural values and norms are with regard to the war on drugs in the Philippines. So it's clear that all the perceptions of maternal stress are being shaped by the level of political engagement folks have with the media, how much women consider politics to impact them directly, and more importantly, the underlying cultural attitude that results in stress avoidance or downplaying of stress. And so to summarize, this is one of the first studies to examine how women conceptualize and embody politics and policies. It also underscores the nuanced attitudes toward the war on drugs and how women respond to the social political stress is deeply rooted in Filipino cultural values and norms. Our findings contribute to the cross-cultural nature of prenatal stress and how the social political climate might impact maternal health during pregnancy, and further exploration of cultural norms to understand women's responses to the perceived social political stress and its effect on pregnancy biology is warranted. So with that, I'd like to thank all the women who participated in my study, my collaborators, my mentors, my funders, and all of my research assistants. Thank you.
Hi everyone. As the title suggests, one of my postdoctoral studies shows that there's dopamine compartmentalization defects which can initiate oxalate-linked Parkinson's disease. So why study oxalate-linked Parkinson's disease? We know in Parkinson, there's degeneration of this nigrostriatal dopaminergic neurons, and especially degeneration process starts in these presynaptic terminals, but we do not clearly understand what happens in these terminals, at an early stage of pathogenesis. Auxin is one of the proteins which function in the presynaptic terminals, especially in clathrin-mediated endocytosis, which is important for synaptic vesicle recycling. What's interesting is it's not just auxin, there are at least 4 other proteins which are involved in clathrin-mediated endocytosis are linked to Parkinson's disease, which clearly says that presynaptic clathrin-mediated endocytosis might have a major role to play in Parkinson's disease. And that's what we explored in this study to really understand mechanisms through which auxin mutations, loss of function mutations, and clathrin-mediated endocytosis defects might lead to Parkinson's disease. And we did that by using, auxin knockout mice. We really looked into, characterized the mice to see if they develop Parkinson's phenotypes, then went into the mechanisms. And our study clearly shows that auxin knockout mice show typical age-related Parkinson's disease-like phenotypes. They don't have any phenotypes, either be behavior, histology, or in terms of synuclein aggregation at 3 months. They start showing these phenotypes 9 months onwards. And what's interesting, and they also respond to L-DOPA treatment, which is a gold standard treatment for Parkinson's disease. So it clearly says we can use auxin knockouts as a model to study clathrin-mediated endocytosis. And that's what we did. We started with a very unbiased way. We looked into whole brain proteomics, and we extracted only the presynaptic terminals and ran proteomics. And what we found was there is dopamine degradation defects in auxin mutants. And that's what we followed in rest of our studies. We saw that there is DOPAC accumulation in 3 months in an early time point, which is an indirect measurement of cytoplasmic dopamine accumulation, which clearly is known in the field that any free floating dopamine in the cytoplasm is toxic to the cell. So this might be one of the ways through which presynaptic endocytosis defects might be leading to neurodegeneration in Parkinson's disease. We did some experiment to really see why there might be cytoplasmic dopamine by electron microscopy and some proteomics. We saw that somehow the synaptic vesicle membrane composition is off, which is making these vesicles not hold the dopamine inside properly. But really, we clearly have to study more on this front. But one last thing we wanted to study in terms of dopamine compartmentalization is really look into dopamine reuptake by DAT. And surprisingly, we also saw dopamine reuptake deficits, and we did some histology and we saw large DAT-positive structures in the dorsal striatum. And we went into ultrastructural analysis, and we saw that it's actually, dopaminergic terminal deformities where DAT is being stuck. So somehow axons make these weird wheel-like structures. It would be very interesting to know why those structures are coming in. Our early study shows that it might be due to synaptic autophagy overload. So overall, that's where we really stand. So due to clathrin-mediated endocytosis deficit, there is this CCV accumulation, misordered SVs, which is ultimately increasing the synaptic autophagy, which might be stressing axons, which is causing this weird axonal deformities. And we also saw possible synaptic vesicle membrane sorting defects, which is ultimately leading to cytoplasmic dopamine accumulation and possibly toxicity. Going forward, I'm going to really go deep into it by also using primary cultures, iPSCs, patient biospecimens to clearly establish how presynaptic endocytosis deficits lead to Parkinson's disease, and take it one notch further to see whether various genetic and environmental risk factors of Parkinson's is leading to Parkinson's disease through the same mechanism. And also I'm interested in studying the impact of aging, because aging is a major risk factor for Parkinson's disease. So study impact of aging on presynaptic endocytosis. Thank you very much. I'm open for any questions. Thank you.
Hi, everyone. The overarching question that defines my research interest is, how is the neuroimmune system built? How is it maintained? But how can it ultimately fail due to aging or disease? Members of my independent research laboratory will ask this fascinating question from the perspective of these sentinel immune cells of the brain called microglia, which play an important role in the development of the brain, in its homeostasis and response to stress. And most importantly, the aberrant activation of microglia is implicated in multiple neurodegenerative conditions. If you take a step back and think about the various functions of microglia, essentially all of these functions have to culminate in lysosomes for degradation or recycling. However, we're just beginning to appreciate how aberrant lysosomal activity can in turn contribute to the development of microglia, their functions, and even some of these pathological states of microglia that we see in these neurodegenerative conditions. Members of my independent research laboratory will seek to decipher this intimate link that exists between microglia functions and lysosomal pathways using the vertebrate model organism zebrafish. I will show you how I intend to use the many advantages of zebrafish, including the feasibility of large-scale genetic screens and live in vivo imaging, as you see on the left here, to illuminate lysosomal signaling pathways in microglia. As a postdoctoral trainee, I showed that the key lysosomal transcription factors, TFEB and TFE3, must be repressed for the normal development of microglia. Prior to my work, it was widely thought that TFEB and TFE3 are essential for the activation of all lysosomal pathways. My research revealed that TFEB and TFE3 do not activate lysosomal pathways under normal conditions, but only do so in response to cellular stress, thus challenging the canonical view of the functions of these transcription factors. The observation that cells have a dedicated conserved signaling pathway to turn on lysosomes in response to stress led me to design the first project of my independent research program to ask, how are signals of extracellular stress relate to lysosomes in macrophages? First, I have identified targets of TFEB and TFE3 in the macrophage lineage, and my first trainees will define the functions of some selected targets in microglia and macrophages. Next, we will perform a genome-wide CRISPR screen in macrophage cell lines to determine how signals of extracellular stress are detected and transmitted to lysosomes both in general as well as to TFEB and TFE3 more specifically. Together, these aims of Project One will reveal the signaling cascade that microglia and macrophages deploy to activate lysosomal pathways. Next, we're becoming increasingly aware that genes associated with the risk of late-onset Alzheimer's disease are enriched in microglia and function in lysosomal pathways. However, the functions of most of these genes in the healthy brain are unknown, and understanding the normal functions of these genes can be key to uncovering how these functions are disrupted in aging or disease. To address this goal, we will identify zebrafish homologs of Alzheimer's genes associated with Alzheimer's disease risk, perform a large-scale CRISPR screen in zebrafish, followed by in vivo imaging of microglia function. We will follow this up with behavioral assays of working memory and executive function in these animals to link microglial activity to neuronal circuits. Thus, Project 2 has the potential to reveal novel functions for disease-associated genes, as well as uncover important and interesting biology of microglia and macrophages. Finally, in my third area of interest, I will ask, are there conserved neuroimmune signatures between pediatric and adult neurodegenerative diseases? Lysosomal storage disorders, which are typically pediatric conditions, are thought to result from lysosomal dysfunction, which in turn leads to the accumulation of toxic metabolites in many cells. These metabolic manifestations are also frequently accompanied by immune reactivity. However, we know almost nothing about the contribution of microglia and macrophages to lysosomal storage disorders, with some exceptions. Using zebrafish mutants for genes implicated in lysosomal storage disorders, members of my lab will perform spatiotemporal analysis of the onset of inflammation relative to neuronal and metabolic defects, and identify small molecules that alleviate disease pathology. Thus, these studies will illuminate the role of the innate immune system in lysosomal storage disorders. In conclusion, today, I hope I've shown you that my independent research program will build upon the strength of the zebrafish system and the novelty of both the approach and the questions I'm asking to lead to a deeper understanding of the basic principles of lysosomal signaling in microglia during development, in response to stress, and in disease. And with that, I will be happy to take any questions.
Hi, everyone. My name is Carol Chua Dominguez, and I will be giving a presentation on titled Social Determinants of Health and the Impact on Adolescent and Young Adult Cancer Survivors and Caregivers. To begin with, I want to discuss how there's been decades of research that has demonstrated that there's a variety of factors that contribute to cancer health disparities, but among the most profound ones that we know impact cancer disparities is social determinants of health, which are the conditions in which we are born with, born in, live, work, and healthcare system. The figure to the right highlights some of the populations here in the US that experience disparities, and my work really focuses on the top three, which are individuals that belong to minoritized groups, individuals who have low socioeconomic status, and who lack or have no health insurance. Additionally, my work focuses on adolescent and young adults. Something unique to this population is that due to their young age in which they are diagnosed, parents often act as informal caregivers throughout the cancer continuum, meaning from time of diagnosis to treatment to survivorship, and they provide a wide spectrum of support, which can often create financial stresses for parents and can cause long-term mental health issues or illnesses such as post-traumatic stress disorders and depression. So some of the quantitative work that I've done is primarily focusing on 2 studies that use population-based survey methods and recruited both childhood cancer survivors and parents. And the study predominantly, or the majority of these participants, were Hispanic or self-identified as Hispanic. And what we found was that both survivors and parents are more likely to seek information from hospital sources. And this is true for parents who, regardless of language that they prefer, meaning whether they're Spanish or English speaking. We've also seen that in dyads where parents are Spanish speaking, they tended to be engaged in more frequent and more comprehensive communication about their cancer experience and follow-up care. Lastly, in another study, we found that childhood cancer survivors' perceived social support was associated with lower parents' depressive symptoms and perceived stress. So what we begin to see here is that the health of one impacts the health of the other. In some of the qualitative work that I've done, recruiting Hispanic parents from LA County USC Medical Center, which is a safety net hospital that primarily sees vulnerable populations, meaning those who have low socioeconomic status and who tend to be underinsured. What we found is this conceptual model demonstrates some of the challenges that they experience throughout the cancer continuum and how that is related to their psychological and financial health, and that leads to caregivers developing coping strategies, which can be summed up or described as in these 3 sort of major themes, which are problem focused, emotion focused, and avoidant. And the most often used coping strategy was problem focused, which included self-efficacy, social support, and behavioral change. In summary, what we've seen is that in addition to a cancer diagnosis, childhood, adolescent, and young adults and their caregivers face issues related to social determinants of health, and that understanding factors that are unique to these Hispanic cancer survivors and caregivers is important. And using this, we are hoping to develop culturally tailored and accessible supportive care services in order to help with the quality of life of caregivers and cancer survivors. I'd like to end by acknowledging participants and staff the institutional and funding support that I've received to conduct this work. Thank you.
Hello, my name is Maria Alejandra González González. I am a research associate at the Neurological Research Institute at Baylor College of Medicine. Today I'm going to present how I have incorporated the use of graphene fiber electrodes to decode autonomic neuronal networks for bioelectronic medicine. Let's define first bioelectronic medicine. This is an emerging field that is focused on the electrical neuromodulation of the nervous system to treat and diagnose disease and injury. This is mainly used for patients that are drug resistant. And the clinical application includes a long list of targets, including carotid sinus stimulation, vagus nerve stimulation, sacral nerve stimulation, and the application goes from resistant hypertension, epilepsy, depression, and the list is growing. The clinical application also the preclinical application also include the metabolism study, heart failure, and glucose control. This is a table that summarizes some of the applications, and we can observe that we have not only the application with the somatic nervous system, but also with the autonomic nervous system and a long list of targets. This is important to understand that the anatomical complexity represents a big challenge in the development of neural interfaces. And here I'm showing a video where we see that the peripheral nervous system are surrounded by fat. And this is the spleen in the rat, where we see that the neurovascular plexus are surrounded by fat. So these are the nerves that are innervating the spleen. If we remember the anatomy of the nerves, we can have a monofasciculated or polyfasciculated nerves that include multiple fibers, including myelinated and unmyelinated fibers. We recently developed these graphene fibers that are high sensitive and also are highly flexible. And we can see this video where we are measuring also the impedance, where we see that these fibers are flexible, we can tie a knot and the impedance is not changing, which is important for neural interfaces. We use these electrodes to implant the neurovascular plexus in the spleen, as we can see in the right. And when we compare with conventional electrodes, we observe that the performance of the recording was really improved in comparison with the conventional electrodes. We were interested in the study of the cholinergic anti-inflammatory pathway. Why? Because in the last decades, it has increased the interest due to the discovery of that by stimulating the vagus nerve, we reduce the systemic inflammatory cytokines. So, but there is controversy. There is controversy whether or not there is direct or indirect connection with the spleen. And we are representing this in this cartoon on the left. So furthermore, when we have the spleen, we see that there is not only one nerve innervating this organ, but also we have different branches that have neurovascular plexus or neurovascular plexus. So the question was, do these neurovascular branches have different phenotype? And to assess this, our objectives and methods were the next ones. First, to characterize the electrophysiological activity in the spleen neurovascular branches by using graphene fibers. Then define the ultrastructural arrangement of the splenic branches by electron microscopy. And finally, use viral tracers for track tracing of this to demonstrate there is direct or not the connection between the vagus nerve and the spleen. So when we go to the electrophysiological characterization, we implanted this graphene electrodes in different branches in the spleen. This is a representative recording, but more interestingly, and it was very exciting to observe that the 4 branches of the spleen in the rat are different, have different activity. And when we reduce the systemic oxygen in the animal, we observe that the response of these different branches also are different. So it is pointing out to a compartmentalization of the activity in this organ. When we analyze the ultrastructure, we observe also that the composition of the fibers was different, which showed that also not only the activity, but also the anatomy is different. We wanted to evaluate the synchronization of the activity with the vagus nerve. And what we did was to implant simultaneously electrodes in the vagus nerve and also in the different branches in the spleen. And what we observe when we modify the blood pressure by administering nitroprusside indicated by NPS here, we observed that the response was different for all of the nerves. However, there was a synchronization of activity with the cervical vagus nerve and the branches in the spleen, but mainly with the splenic branch number 1. So we decided to use tract tracing to evaluate the connection with the splenic nerve 1. For that, we administered adenovirus that express GFP in the splenic nerve 1, and it was exciting for us to see that there was direct connection to the cervical vagus nerve. So we were demonstrating a direct pathway from the vagus nerve to the spleen, corresponding to the terminal branch number 1. So as a conclusion, these graphene electrodes allow to elucidate different activity of terminal neurovascular plexus in the spleen. The differential axonal composition on the splenic terminal branches points toward a non-homogeneous neuronal component through this organ or compartmentalization. And the neuronal tract tracing supported a direct anatomical vagus spleen correction. And of course, we need more studies to elucidate more collateral afferents or efferents to relate to different branches in the spleen. I want to acknowledge all the institutions that have been supporting me in my research and the ISFS that is hosting us today. Thank you very much.
Hello, my name is Arif. I would like to thank the organizers for this wonderful opportunity. The title of my talk is Regulation of Type 2 Inflammation by Diet and Microbiota-Derived Metabolites. We know that microbiota produce hundreds of metabolites, and these metabolites can regulate immune cells either directly or by regulating other cell types like epithelial cells, stromal cells, or neurons. And diet can regulate both the composition and metabolic output of the microbiota. For example, dietary fiber is converted by microbiota into short-chain fatty acids, and this has anti-inflammatory effects. But little is known about the influence of fiber diets on various microbial metabolites. So I wanted to know, how does dietary fiber affect various metabolites and what are the implications in immune regulation? To do that, we chose the inulin fiber, which is very common in fiber supplements, and we used this fiber in mice for 2 weeks, and we performed untargeted metabolomics by high-resolution mass spectrometry. And to our surprise, we observed that there were hundreds of metabolites which were either upregulated or downregulated by the inulin fiber. Here you are seeing 3 different groups. The blues are downregulated and reds are upregulated, and each node is 1 metabolite. So you can easily see that bile acids were mostly upregulated by the inulin fiber. We also did immunophenotyping in the colon. Here you are seeing flow cytometry plots. We found that eosinophils are significantly upregulated by the inulin fiber. And this phenotype was actually microbiota dependent because we did not see this eosinophilia in the germ-free mice. So then we went back to the metabolites and we tested individual bile acids. And as you can see here, single bile acids were sufficient to reproduce the inulin fiber-induced eosinophilia. We also chose a human microbiota strain, Bacteroides ovitus, and we deleted the bile salt hydrolase gene, which converts the precursor into cholic acid. And then when we colonized the germ-free mice with wild-type strain, we observed the eosinophilia by inulin fiber, but that was abolished in the mutant strain. So it is showing that the microbial metabolism is actually driving the inulin fiber-induced eosinophilia. So what is the clinical implication? We know that eosinophils are contributing to allergy. So we used an allergy model, and when we gave mice the inulin fiber, we actually observed exacerbated allergy symptom. On the other hand, the inulin fiber is also given to a murine model of helminth infection because eosinophils are known to have anti-parasitic activity. So as you can see here, the worm burden was actually lower in the inulin fiber group, showing that the increased eosinophils are actually beneficial and protective against helminth parasites. To summarize, I think what's happening here is the bacteroides are being increased by the inulin fiber, and that is leading to more production of bile acids, which subsequently activates stromal cells and group 2 innate lymphoid cells, which I couldn't show you today, which subsequently leads to the eosinophilia and type 2 inflammation. With that, I would like to thank the lab and my mentor, David Ardis, my collaborators, the core facilities, and my funding agencies. Thank you.
Hello, everyone. My name is Roberto Vasquez, and I am a postdoctoral fellow at the University of Connecticut Health Center. And I work with the interactions between oral bacteria and Candida albicans in the context of oral Candida infections. So I will talk about the interactions between diet, oral microbiome, and the Candida albicans infections and the solutions that we are proposing. So oropharyngeal candidiasis, it's a significant health concern, particularly for immunocompromised individuals, but also for healthy individuals. And it is primarily caused by Candida albicans. Oropharyngeal candidiasis, OPC, is an urgent health problem because of the increasing prevalence, the limited antifungal therapies, and the rise of multi-drug resistant strains. OPC is a multifactorial disease. There are several risk factors. Among them, it's diet. Diet can shape the oral microbiome and host interactions. Diet could influence how the metabolisms of the host responds and the immune responses. If from our lab, previous studies show that diet might influence the oral bacteria and the severity of the OPC. So we need novel solutions to combat oropharyngeal candidiasis and other candida-based infections. And the microbiome-targeted therapies is one of these solutions. So what we did in what we have done in the lab, we work with prebiotics and probiotics. For example, cell oligosaccharides, XOS, it's a prebiotic that decreases Enterococcus and Staphylococcus, both bacteria associated with Candida infections, and increase Bifidobacteria, a probiotic bacteria associated with anti-candidal properties. We also found that Lactobacillus jensoniae, MT4, a novel strain isolated and characterized by our lab, is the most dominant bacteria on the C57BL6 mice. And this MT4 strain has antimicrobial properties, particularly against Candida and against OPC-associated bacteria. Also, MT4 can restore the diet-perturbed oral microbiome composition. So when we change the oral microbiome using a diet intervention, MT4 can restore it back. We found that using the prebiotic XOS, we could attenuate the oral mucosal virulence. This is a diet-based therapy in mice. And we found that XOS also prevented Candida-induced oral bacterial dysbiosis. So with that in mind, we further studied the effect of diet. And we found that the physical form of diet also influences the OPC severity. For example, if we have a powder diet, this powder diet exacerbates the Candida infections. Here we have, for example, like a mice under a pelleted diet with two immunosuppressions. We see the fungal biofilm, this white coverage on the tongue. And this is worsened when we have the mice under the powder diet with either 2 immunosuppressions or 1 immunosuppression. Even in immunocompetent mice, mice that have not been immunosuppressed, they develop oropharyngeal candidiasis under the powdered diet. And also the physical form of the diet influences the bacterial dysbiosis, the oral bacterial dysbiosis. For example, we have mice under the pelleted diet, and when they are infected with Candida enterococci, which is associated with Candida infection, it remains kind of the same as the uninfected mice, but under the powdered diet, enterococci biomass increases by several orders of magnitude. So in conclusion, our findings highlight that diet has a profound influence in the context of Candida virulence. We found also that diet can modulate the oral microbiome composition, and if it can exacerbate or attenuate the oropharyngeal candidiasis. For example, we found that using a xylo-oligosaccharides, we can attenuate OPC with the use of antifungal therapies. And this diet-based targeted intervention on the oral microbiome can result in novel strategies for like a therapeutic application and to improve the management of diseases. Thank you so much. I want to thank my current mentor, Dr. Ene Dongari, the ADB Lab, Angela Thompson, our lab manager, and Dr. Tekanari Sobue, one of our collaborators, and Dr. Janja Su from the Zoo Lab at Yukon Health, and Kendra Mas from the Mars Core, and to the NIH for the funding. Thank you so much.
Hi there, my name is Junior West. Thanks again for giving me the opportunity to come here today and share some of my work. I'm very excited to be part of the Intersection Science Fellows. I'm a postdoctoral fellow in Andrew Ewald's lab at the Johns Hopkins School of Medicine. In our lab, we're focused on cancer cell metastasis. This is a multi-step process that's initiated in healthy tissue that has undergone oncogenic transformation and where a tumor will arise. These tumor cells will then eventually gain the ability to invade and disseminate away from the primary tumor, enter circulation, at some point exit circulation, seed into a distant organ, such as the lungs, and then grow out into robust metastases. My work is very focused on studying how epithelial barriers are able to become broken down during this process and how they facilitate metastatic disease. Now, epithelial barriers are mediated by a number of different protein complexes, and my work is focused on studying the tight junction with specific focus on claudin proteins. Now, claudins are these transmembrane proteins that establish the tight junction and establish tight intercellular connections between epithelial cells. In the Ewald lab, we use 3D organotypic culture and in vivo assays involving organoids to test the role of genes in tumor growth and metastasis. So one of the initial experiments that I performed in the lab was to knock down some of these claudin proteins and assess their effect on metastasis. And by doing that, we were able to observe that knockdown of a claudin 7 was able to promote spontaneous metastasis from primary tumors to the lungs in mice. Next, we want to look for a role in invasion, since we know that this is the initial step of metastasis. So we used one of our 3D organotypic assays for invasion to test whether claudin 7 is restricting invasion. And indeed, we were able to observe that loss of claudin 7 induced several-fold increase in cancer cell invasion. We next wanted to look at molecular drivers of this effect. So we performed bulk RNA sequencing of our control and claudin 7 knockdown organoids and observed over 200 differentially expressed genes in claudin 7 knockdown organoids. Of these genes, we were able to decipher a number of different molecular signatures that were associated with these pathways or with this claudin 7 disruption. And we specifically became interested in the specific myogenesis pathway, where we were able to observe an upregulation in a number of genes associated with smooth muscle actin contractility. Staining for control and claudin 7 knockdowns revealed that there was indeed an upregulation of smooth muscle actin proteins, and that this was associated with these invasive cells that are leaving our primary organoids in claudin 7 knockdown conditions. We use a number of different small molecule inhibitors to inhibit smooth muscle actin proteins, and we're able to observe that indeed inhibition of these proteins abrogated cancer cell invasion, indicating that loss of Claudin-7 promotes invasion by inducing a smooth muscle actin contractility pathway. In the future West Lab, I am interested in really understanding how barrier function becomes disrupted during metastatic disease and contributes to increased invasion and metastasis. To do this, we will address three main questions. First, we really want to understand more about the signal transduction pathways that are involved with these Claudin-7 proteins. Second, we want to be able to look at how Claudin proteins actually regulate paracellular transport of cells and different signaling molecules into primary tumors, and how that affects tumor progression and metastasis. And third, we want to establish, define, characterize, and identify transcriptional networks that control Claudin expression that ultimately become disrupted during cancer progression and metastasis. With that, I'd like to take the time to thank the Ewald Lab, my supervisor, Professor Andy Ewald, who's been a great mentor for me, as well as a number of people in the lab who have been super instrumental during my time here in the Ewald Lab. Thank you for your attention. I'd be happy to take any questions.